Now, antibodies are composed of polypeptides with variable and constant regions. You would have heard about this thing. These are identical heavy chains in the greens and light chains in the yellow. Okay? These are polypeptides actually. Heavy chain polypeptides and light chain polypeptides. The light chains are about 25 kilodalton in size, the yellow one. In the green one, they are about 50 kilodalton in size. So a typical antibody, just like an IgG antibody, it would be about 150 kilodalton in size. Okay, the size would be like that much. And there are two very distinct regions. One region that binds the antigen, this is called Fab region. Fab is F is for fragment and AB is for antibody, So this is the antigen binding, and AB is for antigen binding, okay? So this is the antigen binding fragment of the antibody, which is known as Fab, okay? And this one, the lower portion, this is called FC, or crystallizable fragment. Okay, our fragment crystallizable, FC portion. So the FC portion is not binding with the antigen, but the other immune processes that I just told you about, like opsonization, antibody-dependent cellular cytotoxicity, complement activation, for instance, all these things are actually this this portion of the antibody is responsible for those functions. For instance, this FC region has got FC receptors on many different immune cells. You would have heard about the FC receptors, for instance, on mast cells and other cells. Okay, so this FC region of the antibody is going to interact with other immune cells and help with the functions of other immune cells. So all other functions of the immune system, including opsonization, complement activation, ADCC, etc., are due to the FC region of the antibody, while the Fab region just binds with the antigen. Okay? So this antigen binding site actually is in the variable region of these peptides. If you look here, this is the heavy chain, the green one, this is the light chain, and if you compare it with here, the topmost portion in both these cases, the heavy and light chains, they are the variable regions. Okay, there is variation of sequences in this region. That's why we call them the variable regions. So the variable region in the red, it is actually the antigen binding site. And the blue one that you see here is called the constant region. Okay, so variations in the constant region are less frequent. Variations in the antigen binding sites, these are more frequent, and that's why this variation is needed as well. The diversity of this specificity, as I told you, 10 raised to the power 9 different specific antibodies can be produced by the B cells. Okay, so that is because of this variation in the amino acid sequences of the variable light chain and heavy chains variable regions. So these genes, they undergo a lot of variations due to this process of recombination. When the genes are recombined with each other, you would see in the later slides, and that would create different specificities, means that different combinations of these variable regions are these polypeptides, which can be complementary, different antigens, different types of viruses, different types of bacteria or other ligands, okay, whatever enters the body as an antigen, there would be a specific antibody for that antigen as well. And there is a hinge region right here, okay? And this is also very important. You would see in the next slide why this is important. But just in this slide, you remember the heavy chains and light chains. Amino terminus is here. The antigen binding site is amino terminus or the N terminus of this antibody. While the carboxy terminus, it is relatively conserved region or the constant region at the carboxy terminus. Okay, and the total size of the antibody is 150 kDa with these different regions that you see here. Now, if you see here, this is a bacterium, and as I told you about the hinge there, if you see here, this hinge, it has different orientations in the case of different antibodies attached to this particular bacterium, okay? Because on the surface of the bacterium, you can find different types of epitopes. Epitope is a region or, antigenic determinant on the surface of a pathogen that can bind to an antibody, okay? We call it an epitope, okay? So if you look here on the surface of this bacterium, there are different types of epitopes given in the red. These are antigenic determinants, and the antibodies are going to bind to all these different determinants, because for all these determinants there are specific antibodies in your system, in your body. Specific B cells in your body are there, okay? So these antibodies can be, these antigenic determinants can be in different variations. You see, some of them are straight and some of them are curved, with different topologies. So the antibodies are also flexible here due to this hinge that you can see here. They can just open up a little bit, or they can just, close down a little bit, or they can just straightly go there and fix to the surface determinants. So these carbohydrates, glycoproteins or glycolipids, etc., these can be of different nature in different shapes or different topologies, and for all those things, these hinges actually are helping with the antibody to bind very specifically there, just like a lock and key mechanism that you already know about. Okay. Now there can be a single antigen with multiple epitopes, and that multiple epitope can be, there are different epitopes as you see here on the surface of this bacterium. One is in blue, green, and yellow, etc. Some are round, some are, rectangles, and some in different shapes. So for all these different shapes, you see that the antibodies binding to these multiple epitopes, they are utilizing different, options. For instance, you can see there the antibody has conformed itself to, the shape of the antigen or the epitope. Okay, so they would just go there, and different kinds of, strategies would be used. Or different antibodies can attach to the same, antigen with different epitopes, because the nature of these epitopes can be different, Similarly, a multivalent antigen with a repeated epitope can be bound by the same antibodies, Same antibodies can just surround these epitopes if they are similar epitopes on the surface of a pathogen. But usually on these surfaces, like the surface of bacteria, etc., particularly these microbes, you can find many different types of epitopes recognized by many different types of antibodies. And that's why multiple antibodies of multiple specificities are involved in, tackling that particular antigen or, that bacterium or virus, etc. And this binding that you see here or in this slide, this binding is based solely on non-covalent forces, and these are the non-covalent forces given here. Electrostatic forces, hydrogen bonds, van der Waals forces, and hydrophobic interactions, that help the antibody to bind with epitopes of different antigens. Now there's a little bit detail of these different regions. The constant region, amino acid sequences in the constant region, terminal regions of the heavy and light chains is the same. These are the constant regions. If you look at the antibody, this is the light chain, this one is the light chain, okay, and this one is the heavy chain, okay. Now it is further, divided into sub-regions. For instance, in the light chain, you can see that there is a variable light region and there is a constant light region. Similarly, in this, heavy chain, you can see that VH is for the variable heavy chain, and there is a constant heavy chain. Okay. Now, the variable light chains and variable constant light chains. You can see that there is a single variable light chain segment followed by a constant light chain segment. In this light chain, you see VL and CL. But here, in the case of the heavy chain, you see that there is a single variable heavy chain that you see here in the case of this polypeptide or this one, and there are multiple constant chains, constant segments. Okay. For instance, CH1 is the constant heavy, chain 1. This is constant 2. This is constant 3. So there can be variation in the number of these constant segments. In case of different antibodies, this constant chain is the number of these segments is different. And the variable chain amino acid sequences in the N-terminal regions of the heavy and light chains is different. As I told you, this is variable of a region of the light chain. This is variable region of the heavy chain. Okay, and amino acid sequences are quite different from the rest of the antibody regions. And this region provides antibodies with unique specificity, as I told you. Only the variable, specificity lies only there at the variable region. And that provides the antibody with this specific arrangement of genes that is responsible for binding with different pathogens. Then there also is a hypervariable regions that I will discuss in the next slide. In these VL or VH domains, there is another very specific hypervariable regions. So actually, the actual binding site for the antigen is that hypervariable region. Okay? So this is a broader region, variable region, and then there is a specific region which is known as the hypervariable, where the variation is actually is found there in that region, and that region is actually the antigen binding region. Okay, what do you see here? Apart from that, you see that This is the heavy chain, and this one is the light one, and both of them are attached with each other through disulfide linkages. If you see here, the heavy chains in themselves also are attached with each other through the disulfide linkages. On both the sides, you can see the same pattern repeated. Okay, so these are covalent linkages that are actually linking the polypeptides here with the polypeptides here. Okay, the light and heavy polypeptides, all these polypeptides are joined together, and their segments join with each other through the disulfide linkages. So these are covalent linkages. But when antibody binds to a particular pathogen, that would be either van der Waals forces, hydrogen bondings, these electrostatic interactions, okay? And that binding would be there, along with the variable region that we are talking about, okay? This variable regions contains lots of these aromatic amino acids, okay? And these aromatic amino acids, they provide for this opportunity to make these, uh, you know, uh, unstable bondings with the antigens in the form of hydrogen bondings or other electrostatic interactions, okay? Now, if you see here, what are the heavy chains types or isotypes? There are different heavy chains isotypes that you see here: mu, gamma, alpha, okay, epsilon, and there are only two light chain isotypes. One is kappa and the other one is lambda. Okay, for this there are only two isotypes, and for this chain there are multiple isotypes. You know, so if you look at the significance of these isotypes, these multiple isotypes, they usually would remain the same in case of different antibodies. But if you look at the light chain, kappa and lambda, these isotypes in different antibodies, this can either be a kappa chain or this can either be a lambda chain. Okay, but never both. In the case of a particular antibody, you would not find that one chain is kappa. For instance, on the right side, it has a kappa polypeptide, and on the left there is a lambda. It can either be kappa in kappa or lambda in lambda in the case of different antibodies. Okay, and this, if you look at the prevalence of these kappa and lambda, again, there is relative abundance of these in different species and different animal species. For instance, in human beings, a majority of the antibodies, they have a kappa light chain, okay? And minority of the antibodies, they would have a lambda, but in majority cases you would find the kappa chain in human bodies. If you look at the length of this fragment, it's about 214 amino acids, and the heavy chain, it's about 446 amino acids in length. Mm-hmm. I will tell you in the next slide maybe, because it's coming up. Okay, then I told you that in the variable domain, there is a hypervariable region. Okay, these are the variable domains that you see here at the top in different colors. This is again a different representation of the same thing. This is a heavy chain and this one is a light chain, and those two are the variable regions. So the hypervariable regions here is found in case of the light chain and the heavy chain. Both of them have hypervariable regions. 3 hypervariable regions at least, In the epitopes are the antigenic determinants on the surface of the pathogens bind specifically with this region of the antibody, the hypervariable region, okay? So within the variable domains are 3 regions of extreme variability that are called the hypervariable regions. And these regions of the variable domains actually contact the antigen. They therefore make the antigen binding site. फैब रीजन में ये एक हाइपर वेरिएबल रीजन है जहां पे ये एंटीजन बाइंड करते हैं. These regions are also called the complementarity determining regions or CDRs. इनको CDRs भी कहा जाता है. Okay, you can call them CDRs. Okay, so if this is hypervariable region 1, 2, and 3, you can also call it CDR1, CDR2, and CDR3. Complementarity determining region, because this region determines complementarity with what? With the antigen. Okay, with the epitope or antigenic determinant that is present on the surface of a bacterium or a virus or other microbe. This region actually is going to be complementary with that particular peptide. Okay, if it is not complementary, they cannot bind with each other. So this region is going to determine complementarity. The type of bonding that I told you, that whether these electrostatic interactions would attract a particular, antigenic determinant or epitope, which would be a best fit in that situation, or in terms of energy dynamics. Everything cannot bind with an antibody. Non-specific things cannot easily bind with an antibody because of the energy dynamics that are created here at the micro level. We are just talking about a 150 kDa molecule, okay? And we are talking about an extremely small micro environment where all these things are happening, and the bonds that are created there as a result of these interactions. So if it is not a best fit, it would not bind to this region. This is complementarity, okay, which is decided here when these antigens come around an antibody, okay. So you see that there are different domains attached with each other through these disulfide linkages, but these, different domains, the length of these domains is about 100 to 110 amino acids. We call them immunoglobulin domains, okay. These are compact domains, and these domains, also confer stability to the antibodies, because anything can become unstable inside the body in different situations, because the antibody has to work outside in the tissues, and there are lots of proteases that can cut this antibody into pieces. There are lots of other things that can change the pH of the environment where the antibody has to work, okay, and make the pH of that environment less suitable for the antibody to be active or to work, okay. So what would the antibody do in that case? The antibody has to, if the antibody is destabilized, the antibody would not work, okay. It would not work, or the intended job will not be performed by an antibody if it is destabilized due to any situation. If it is destabilized due to pH, or if it is destabilized due to any other reasons like the presence of different enzymes or different proteases that can, cut this antibody into pieces. So this stability is provided to the antibody with the help of these immunoglobulin domains. They just, fold around, okay, these 110 amino acids domains, they would just fold in a way in order to make a stabilized structure, okay, and that stabilized structure is protected from being degraded by these different enzymes, or it would remain active in all situations. So this is the natural protection of an antibody that you see there. Now the antibody, if you see here that there are different types of epitopes, as I told you in the case of different antigens. You see here, this is one type of an epitope or antigenic determinants found on the surface of the pathogen. There is another type of antigen. You see here is another type of antigen, and this ball is another type of antigen. All these antigens are recognized in different parts of this variable region. The variable region is the binding site for all these different types of antigens. But the binding is not the same in all the cases. Okay? For instance, it can, these epitopes can be trapped here. You see, this is a small type of a pocket-like thing, like you hold an apple or something in your hand. I'm holding this particular thing in my hand, okay? So for sugars, for instance, there are lots of sugars on the surface of microbes, and for sugars, usually in the hypervariable region, there is, the formation of the binding site would be just like a pocket, and this sugar would be bound with the pocket. If there are multiple sugars, then this binding site can just modify into a group to attach with multiple, uh, you know, uh, sugars together. If it is a protein, for instance, this one is a protein, then an extended surface is used. Rather than going for a groove or a pocket, this whole variable region that you see here at the top, this would be used as, as a whole, okay? So that surface would be used, an extended surface of the variable region would be used for a protein found on the surface of these different microbes, okay? But in certain cases, the nature of the antigen would be quite different, just like as you see here in the form of this red ball, okay? And for this red ball, this hypervariable region has another option. This is the knob-like structure, and rather than the variable region or the antibody making itself to fit with the antigen, the antigen has a particular structure where this this variable region is going to fit into, just like a knob, okay? So this is another form of the antigens. In case of different types of antigens, the purpose of all this that I'm saying right here, that in case of different antigens, which can be, there can be different types of antigens on the same microbe or on in case of different microbes, the variable region can be modified according to the nature of that particular antigen, and it would be, it would attach with uh, the variable region, which is responsible for the function of the antibody. Just about the isotypes or classes of antibodies, you have heard about 5 different classes of antibodies. We call them IgG, IgM, IgD, IgA, and IgE. Okay? These are 5 different classes of antibodies. Okay? Now you see here, some of these antibodies are secreted or in the form of monomers. For instance, immunoglobulin G is secreted in the form of monomers. Some of these antibodies are secreted monomer and dimer, just like IgA. But you see that majority of these antibodies, IgG, IgD, and IgE antibodies, these are secreted as monomers. Okay? And only IgA is secreted as monomer or dimer, while the IgM is secreted as a pentamer. Okay? There will be 5 different IgM molecules attached to each other to make a pentamer. Okay? So now if you look at the structure of these immunoglobulins or 5 different classes of antibodies, do you see any difference in the structure of these antibodies?
Yes.
What is the difference?
There are more segments in the constant region and active.
Yes?
There are more bicycled with the IgM and the IgE.
Yes?
The sugar attachment.
Yeah, the sugars, these are the sugars attached to these chains, you know, the constant regions.
And the constant region, the number of points in IgM and IgE is different.
Yes. So the constant regions are attached with carbohydrates. You see, the number of carbohydrates is different in each case, in the case of each class of an antibody. You know, the nature of carbohydrates and the number of carbohydrates could be different in these cases. You know, then you see that there are, if you look at the constant region here, how many segments are here? Domains in the case of IgE, 1, 2, 3, and 4. Another one is IgM here. How many domains? 1, 2, 3, 4. Okay? 4 domains and IgM and IgE. Okay? But the rest of the antibodies, you can see that they have only 3 constant regions. CH1, CH2, CH3. Okay? But IgM and IgA, they have an extra domain, which is CH4, constant region 4. Okay? Another difference is, just look at the shape of this antibody. In this antibody, there is a clear difference between the two. Other than this difference, the number of constant domains, there is no hinge in IgM that you see here. Okay? And there is no hinge here in the case of IgE, but all other antibodies, they have these hinges. Okay? Now, these are the names here. The heavy chain of each isotype is denoted by their corresponding lowercase Greek letter. Gamma, for instance, here. This is a gamma chain, mu, delta, alpha, and epsilon. So these different classes of antibodies are different from each other because of this constant chains, the constant chains that are attached there are that are part of these antibodies. Okay, the constant chain in case of immunoglobulin G is lambda, immunoglobulin M is mu, IgD is this one. What is this called? Delta. Then this is alpha and this is epsilon. So these chains are different in case of these antibodies. That's why we give these antibodies different names. They are different antibodies. And these antibodies are produced in different situations. You know, the B cells usually would have IgD or IgM as part of its membrane. But when it comes in contact with the antigen, you know, or it is influenced by T helper cells, some cytokines, then there would be class switching. You know, the class switching is, you know, this is the class 1 to class 5. So different cytokines can help the B cells to switch from one form to another form. For instance, if this is an initial stage of infection, an IgM is produced by the B cells, and then after some times IgM production is stopped, and then the B cells need to make another antibodies, just like the IgG antibody. Then the class switch would be needed in case of chronic infections that would persist for longer period of times, or infections that already have resolved, So in those cases, you would find IgG antibodies in the system, not IgM antibodies. These are found in acute infections, but not later. Okay, so this class switch can help the B cells to produce a different kind of antibody according to the situation. For instance, a B cell can be influenced by interferon gamma secreted by a T cells to switch from IgM to IgG, for instance, or some other interleukins like interleukin 4 or interleukin 5. They can also help with class switching, influence the B cells to make a slightly different antibody. So the memory cells that are going to form, for instance, the memory cells, what kind of antibody would the memory cell make? Can you answer this question? The memory cell that was formed as a result of infection. I should rather have given this question in the paper. What kind of an antibody would be produced by the memory cells? IgG. Yes, why? Uh-huh. Okay, you have to study about that. Okay, but why the memory cells were produced IgG? Okay, in the next class you have to tell me that why does it happen actually. So if you look at the abundance of these antibodies in the circulation or normal serum of the patients, majority of these immunoglobulins are IgG. Okay, and other immunoglobulins are also found, but relatively their abundance is less as compared to immunoglobulin G. Okay, so in the next class you have to tell me about the class switching, that what kind of cytokines are responsible for switching of class by the B cells to form another type of antibody. Okay, you have just, this is just a short assignment. You should not write many things. You can just draw a diagram and just explain that. That's enough for today. I think we can just have